A simple stain uses only one dye or chromophore. In a direct stain, a positively charged chromophore colors the negatively charged cell. The purpose of a simple stain is to allow the visualization of very small, clear bacterial cells. This stain provides information such as the relative cell size, shape, and arrangement. Prepare and fix a smear. Be sure the smear has completely air-dried before fixing. Heat fix a smear by quickly passing the slide through the flame 2 or 3 times. Cover the smear with methylene blue for 30 to 60 seconds. Be sure to cover the entire smear. You may need to use 5 or 6 drops of methylene blue. The amount of methylene blue and the staining time may vary with the reagents used in your lab. Wash the slide carefully with distilled water. Tilt the slide and aim the water above the smear. Let the water wash over the smear. Gently blot the slide to dry it. Do not wipe, just gently blot the slide. Make sure that the slide is completely dry, then examine the slide under the microscope. The type of cells that you will see on your slide depend on the type of culture used to create the smear. When you have finished observing your results, gently blot the oil from the slide and store it in a slide box.